Welcome to part 2 of changing allele frequencies. So we're going to move on to the last two violations of Hardy-Weinberg. So mutation, so change in DNA. And allele frequencies can change in response to it. Mutation introduces new alleles, can convert one allele to another. But keep in mind, mutation can have really minor impacts. Most are actually silent, don't do anything. They're not actually in a gene itself. And even if it does change something, whether it's for better or worse, unless it's coupled with another effect, such as a small population size, natural selection, something like that, it can take a long time before mutation really impacts or changes a species. On our Lucky Charms graph, we've got the blue triangles and green diamonds becoming yellow squares, so those traits will increase and the other ones decrease. All right. Now keep in mind there can be good and bad mutations. Selection generally will eliminate the bad alleles. Dominant, they disappear pretty quick. You have the disease, you die, you don't reproduce. Recessive ones can take a while, though, because they're only seen when somebody who's homozygous recessive appears and fails to reproduce. Sometimes when you talk about a population, you'll talk about its genetic load. So this is just the number of recessive bad alleles that's present in a population. All right, and then finally we got natural selection. So basically there's a differential survival and reproduction of individuals of a certain phenotype. You can either have an increase in natural selection, there's a positive selection for that trait, or a decrease or negative selection for a particular trait. And then you can see that can create changes in the population as a whole. So if the yellow squares can't reproduce, of course, they're going to die out really, really quick, and you'll have no more yellow squares. So we see natural selection among the bacteria and viruses quite a bit. We've seen in TB, tuberculosis fairly often. It's probably one of the largest all-time killers of humans. Flu will actually kill more people in a year, but over time, tuberculosis probably beats it out. We have seen a lot of them becoming antibiotic resistant, so that's been a problem. We've also seen with AIDS, those people have a harder time fighting off TB infections, and so we find more and more bacteria that resist the antibiotics due to being exposed to them more often. The ones that survive are the ones that are resistant to the antibiotics. So there are two ways that bacteria become resistant. One is it's actually a mutation in its chromosome, if you want to call it that, its nucleoid. So that can be, and then it's just passed on with the division of the cell. Another way you can get it is bacteria have little genes known as transposons, because there are these small little plasmids or small little brown DNA things, and they can actually pass this to another living bacteria. So it's a way that they can quickly disseminate resistant genes through that. So then we find all these resistant bacteria like MRSA, which is methicillin. It's a very powerful antibiotic, resistant Staphylococcus aureus. So Staph aureus is the most common bacteria probably found in teenagers, cause a lot of skin infections and things like that. And many of them are becoming resistant. Viruses, we also see natural selection in, even though technically viruses aren't alive, but they do mutate. And when they do mutate, those repairs aren't made. There's no way to fix a mutation in the DNA. So viruses can accumulate mutations very rapidly. And we see this quite a bit with HIV. And as the diversity of these variants increase, we'll see the disease actually progress, because basically the virus is working to tailor itself to be better at killing you is what's going on. The selective factors for the HIV virus is the human immune system and those different drugs that they use to destroy viruses. So only the viruses that are good at resisting, hiding from your immune system, and are not destroyed by those selective drugs can survive. And those are the ones that hang around and are very good at killing you. So the combination of these, what they'll do is they'll give you a combination of drugs. They'll take one drug off, add another one, give you two. So this way they're always finding something that's hitting those viruses to prevent this kind of spread, this change in the virus. So if we look at symptom severity versus viral diversity, so when you get the HIV virus, corona would actually work in some of this ways too. When you first get it, the symptoms are very severe. But then your immune system ramps up, starts killing the viruses. As the virus, though, is replicating, only the ones that can survive in you are going to survive. The other ones will be killed off. So you're going to see the symptom severity fall after a while. But then the viral diversity is getting, actually it starts getting very, very high. But then it's going to fall because only the ones that your body can't kill off or the drugs are taken care of are going to survive. So then it gets very low again. So this virus right here is very low diversity, very little change in it. But because your body can't protect yourself, your symptoms are very high at this point. So then you're going from basically your immune system break down and you're going to die of the AIDS syndrome. All right, one more thing we need to talk about, and that's balanced polymorphism. So there are some alleles that are hanging around, and they're found much more commonly than they should because there's something beneficial by having it. So there's something good by having a bad allele, usually in the heterozygotes. The heterozygote will have one good allele and one harmful recessive allele. There's basically two forces pulling at them. The allele is harmful. If you got two of them, that person's going to die of having two genetic disorders. But if you only have one, it might protect you from some environmental threat. So that's going to be, so there's this beneficial and harmful thing going on. So it's pulling in two different directions. Basically, this is called the heterozygote advantage. The heterozygotes survive in environmental conditions that neither homozygous person would survive. Let's look at some examples of that. There's a number of them. Sickle cell, though, is probably the most looked after. Sickle cell disease is pretty bad. If you've got two recessive traits, with modern medicine, we can average you to about age 40, but a lot of people die much younger than that. Malaria can kill you as well. So you got two things that can kill you here. Anyway, the beta hemoglobin is the gene that this is in. Kind of talked about this earlier in the quarter. So the sickle cell allele is recessive in that you won't have the disease unless you have two of them. But it's really a codominant gene. So if you have one copy of the gene, you're going to make some of these bad hemoglobins, beta hemoglobin proteins. But you also have a good copy of the gene, so you're making roughly in nature about 60% of your hemoglobin is going to be good, 40% is going to be but having two copies of the trait gives you sickle cell anemia, and that's a negative selection. But malaria seems to be killed by this. The malaria protozoan lives in your blood cells. Having sickle cell, apparently malaria doesn't survive very well. At least the blood cell that it's hiding in won't survive very well. As the malaria organisms taking nutrients of the blood cell, it causes the blood cell to sickle, and your body will remove it and remove the malarial parasite. So that's a positive selection. And if we look in Africa, where malaria is particularly deadly, and we notice where we find sickle cell anemia and where we find the malarial organism here, you can see there's a lot of overlap in the gene and malaria. So that's balanced polymorphism, good and something bad from the gene. Yeah, believe it or not, if you're heterozygous for the prion disorder, you can be protected from diseases you can get from cannibalism. So if you're heterozygous, feel free to be a cannibal. You're not going to get the spongiform encephalopathy that can occur from that. I think we've discussed this in the past. Of course, I'm being very sarcastic. Don't eat your own species. Cystic fibrosis, probably the most common genetic disorder among people of Caucasian descent. Turns out having one copy of this gene can protect you from diarrheal disease. Believe it or not, diarrhea diseases is the major killer in the world. It kills more people than anything else having diarrhea. Due to dehydration usually. And having cystic fibrosis actually protects you from some of these diseases that cause diarrhea. So cholera actually goes through the chlorine channel that has been mutated. So it's been mutated, so there's not as many of these chlorine channels to work, and therefore cholera can't cause the severe dehydration that it normally would. Typhus, same sort of thing. It can't enter the cell because it requires a functional chlorine channel. It's not working. It can't get in the cell, so therefore it's not going to survive. So it has a lower survivability. Of course, you need some of these channels working, or else your lungs get full of all the dust and gunk that you're breathing in. It doesn't get washed out. But if you don't have as many, you don't get the diarrhea, and you still can wash out your lungs. But being heterozygous for the disorder seems to offer you protection from diarrheal diseases. And I believe that ends the notes that we wanted to do today. All right, so now you'll have your quiz on basically the Hardy-Weinberg. How does it keep things from changing, and how does it allow things to change? So what's going on there? So that ends these notes.